works. Well, I was mentioned before, this is really, truly a teamwork. Number of people at the University of Minnesota. Mike made a presentation earlier today. I listed Shan there only, but in his group, but he has a tremendous group of people working on the basic aspects of PRRS virus. Bob Morrison and his group work on the swine health monitoring program. And myself, I had the pleasure to work with a number of people focused mostly on data analysis and all the analytical and connectivity aspects of the data and the information. And then acknowledge the project participants. The swine health monitoring program was mentioned earlier today. Actually, 5 participants that share their data, among those in the swine health monitoring program, to run the studies that I'll show in a minute. And then our regional control program, North to 12 in Minnesota, and the program participants and the program coordinator, and the number of sponsors that helped us with funds to conduct the research, including BI. It was mentioned in the previous presentation, number of measures that are combined and then applied to control PRRS at the farm level. When I was in Davis a number of years ago, I got Dale Paulson, Enrique Mondaca, Jean-Paul Cano, who was with BI at that time, paying me a visit and chat about the things that we were doing in a number of countries on foot and mouth disease, and we talked about PRRS and how those things may be applied. And can't remember who was at that time who said that, but I'm going to quote the 3 of them, that the strategies to control at the farm level perform reasonably well, but we still need to understand how to control the disease at the regional level. And that was how I got involved into PRRS a number of years ago, may have been 2007, 2006, and that's going to be the focus of this presentation today. And what we have been doing over the last couple of years, was to try to develop methodological frameworks to evaluate progress of regional control programs. That's going to be study 1 in today's presentation. And second, to identify the emergence and monitor the spread of new PRRS strains. I'm going to build on some of the things that Mike already presented earlier today. So to get into the first study, this paper that has been submitted is already been reviewed. Hopefully it will be out soon. But it's actually Pablo's work here that I'm going to present. He's one of our graduate students. Using N212, this regional control program, what we tried to do was to evaluate or to develop this methodological framework to evaluate how well we are doing. So for those of you that may not be familiar, PRRS is not reportable in the US, so a number of regional projects have been conducted in different parts of the country in which producers voluntarily share information on how they are doing, incidence of the disease, and certain other information that may be valuable for others to control the disease at the regional level. Now the question is how we evaluate how we are moving forward with those programs. Are we making progress? Are we improving or not? And we divided, Pablo divided actually this task in 3 different objectives. First, to analyze the demographics of enrollment, so how producers enroll into the regional program. Second, the demographics of the active participation, so actually they have enrolled, but are they really sharing information? How they are sharing the information? And then finally, the trend, temporal and spatial distribution of incident cases as they progress over time. The data that we use roughly corresponds to the northern part of Minnesota, and we collect information at the farm level. This is a longitudinal study. We analyzed the last 2 years of data from June 2012 to June 2014. We got the locations, latitude and longitude of each of the farms, date of enrollment, PRRS status collected weekly, and then type of farm. So 3 objectives. First objective, how we assess the demographics of enrollment. And what we did here was to divide our data sets in type of farms according to where they had sows or not, just to simplify the analysis, that were enrolled in the program. Then we ran an ANOVA test where the null hypothesis was that the proportion of sites with sows and without sows enrolled in the program was constant through time. So that was the null hypothesis of no evolution, no change. The program is stable and maintained the same throughout. Then the second analysis was to assess the demographics of the active participation, the demographics of sharing. And here we fit a generalized linear mixed effect model for a binary response where the response variable, what we are trying to model, what we are trying to assess, is whether sites were sharing or not their information among those that were enrolled in the regional program. And then we compare that with what we call the fixed effect, so the variables that we think are related to that probability of sharing, where we included time and type of farm. And then we wanted to account or control for a number of factors that we think may influence that response, but we are not really interested in, like the county in which those farms were located, or the site specifically. And we included those values as random effects. And then we assessed the spatial and temporal probability of sharing at the regional level using a test called normal scan statistic. And then finally, we assessed the temporal and spatial distribution of Perez incidence. And again, we ran a generalized linear mixed effect model for a binary response where the response, in this case, was Perez status, positive and negative over time on a weekly basis for each farm that was part of this program. And then this time as effects, we included time, the type of farm, the probability of reporting Perez, which was fitted from the previous model, farm density in the county, proportion of vaccinated farms in the county. And then we tried to assess if those variables affected the probability of having Perez. And then, again, we accounted for a number of factors that may influence the result, but we are not really interested in, like the specific site from where the data were collected or the specific county. And then we also assessed the time-space correlation of pairs of incident cases. So we wanted to see what was the structure of the spatial and temporal conditional dependence of the data of incidence cases. So these are the results. First, demographics of enrollment. So what we see here is that in N to 12, the number of farms enrolled and the geographical coverage increased, but the ratio of sites with south and without south remained constant throughout, basically keeping a relation 1 to 3. So one site with south to 3 sites without south, which more or less resembles the distribution in the region. So with this, what we see is how it increased slightly in time, but the proportion remained constant of sites without south and sites with south. Then the demographic of sharing, the participation increased significantly. So remember, here what we are assessing is actually how many of those that were enrolled were actually sharing information on a weekly basis. And we can see that interesting increased the active participation in the system, but sites without south were less prone to report than sites with south, although it improved in time. As we can see at the end of the period that we assessed, these two proportions were becoming similar. So what the blue line is showing is the proportion of enrolled sites that were actually sharing information on a weekly basis, and this is sites without south. And then we also identified that the variability of participation was higher between than within counties. So there are some counties that were more likely to report than others. And then this is showing the spatial distribution of that participation. And basically we are using this technique to assess within the regional program where we have the higher probability of reporting and sharing information, which basically occur in this region. During the second half, there was a higher interest in sharing information and data here during the second year compared to the previous year, the first year. And then finally, we assessed trends. What we found was a significant decreasing trend on Perez incidence over time. Interestingly, we found also that the probability of sharing Perez status was negatively associated with Perez incidence. So the more they share information, apparently was associated with a reaction on the incidence. Now, it doesn't imply causality, right? Could be you can make this interpretation on two completely different ways. It may be that because they are sharing, they are being more successful in controlling the disease, or could be that they are more likely to share information when they are negative, right? But it's still interesting that we identify this association in this program. We also found that density of large and medium-sized sites in a given county was positively related with Perez incidence. So the more the density in the county, the higher the incidence, which is something that we have seen in the past, but it's always good to see again, because this reinforces the quality of the results. And then we didn't find an association in this study in the proportion of vaccinated farms with the disease, but vaccination was modeled here at the county level, so there may be a bias in that analysis. And also, it could be that the effect of vaccination may be observed in a cumulative fashion, and we were analyzing only 2 years of data. So it could be that we failed to identify the association when perhaps it was there, but we couldn't find it in our data. This is showing the spatial and temporal distribution of pairs of incident cases. So it's showing an increment in 2 peaks of cases in the PRRS, and then density map showing where most of the cases were located. This is showing, actually, there was a funny thing here with the red peaks here. Well, ignore the red peaks here. But it's showing that despite the incidence was going down, the spatial and temporal aggregation was still constant. So most of the cases tend to occur in this region within 3 weeks and within spatial windows of 3 kilometers. And that's repeated over time, even though the incidence overall is going down. So, take-home message for this first study. We follow a 3-stage systematic approach to evaluate the evolution of this regional control program. Evaluate enrollment first, evaluate sharing second, and incidence third. The first 2 metrics are more related with the social aspects of the disease and the participants. The third one is more related with the epidemiology of the disease itself. In this particular assessment, we found that farmers' enrollment was not necessarily a good estimate of participation. They may be enrolled but may not share information on disease status. Sharing information increased, and the proportion of sites without sows were less likely to report and share information than those with sows, although it improved over the last year. As we said before, interest in sharing information among producers was negatively correlated with disease incidence. This is a very interesting finding. I still need to find out on what's the biology behind this association. And then finally, Perez incidence decreased, but spatial and temporal aggregations remained over the study period, which is also interesting. So overall, the incidence is going down, but the spatial and temporal patterns are still the same. Future plans for this study? Well, we would like to apply this systematic approach to our regional programs, and looking forward to working with other regional programs that may be interested on doing a similar assessment, and then compare if this is replicated in different regional programs or there are differences in different locations. And then as a follow-up, we're going to evaluate what are the social and economic factors that may affect the participation in the regional control program, and what may be the collaborative strategies to control peers at the regional level. So this is coming from social science. And Pablo he's a student working on this project, finalizing his master's in economics and doing PhD on this. And the theory here is that as a group decision, and there is complex dynamics in the relation of these individuals. So what benefits myself may not benefit others in the region. And then where and how we reach the equilibrium, how my actions may benefit the regions, and how this combination of actions may actually improve the region when sometimes some people may be benefited by an action that doesn't benefit the others. So that's called game theory in economics, and basically look at the farmers and producers and practitioners as players in this game, and then try to identify where the equilibrium is, how we can motivate those actions that will benefit the group and not the individuals alone. Then the second study, Mike already helped me making presenting some of these slides. This paper is also has been submitted and is going under review, and hopefully will be published soon. Will be out soon. The objective here was to propose a methodological approach to monitor the emergence and spread of PRRS strains. And Mike already made some comments on this, and there were some discussions, but just to back up a little bit, this is a simplified version of PRRS sequences. So suppose that each of you have gone through a number of farms and then have collected a number of sequences, one sequence per farm, and you end up having, and each of these blocks here are a simplified version of the sequence. And then each of the boxes here may represent an amino acid or a nucleotide, right? So we have these sequences that are different from each other. The next step, what we do is a process that is called alignment, in which basically we compare the sequences and we align them, and now we can compare the composition of the nucleotides in each of the equivalent positions. So each color here represents a different nucleotide. There are 4 colors. And then we can compare each of the sequences on their positions. There was an interesting discussion before about recombination. We're not talking about recombinant strains here. If we just compare those sequences, we come up with what's called a matrix. And in this matrix of nucleotide differences, basically I can compare each virus with all the other viruses in my sample, and then I can count the number of different nucleotides that I have in this sample. And then with that information, we compute what's called the genetic distance, which is not only, we account for a number of things. It's not only the proportion of different nucleotides. There's some adjustments done there. But if you want to look at it in a simplified way, you may look at this as a probability of being different. And then probability of finding one nucleotide difference when 2 strains are compared. And then with this information, if I get 1, then that basically means that they are totally different. Sometimes we compute the matrix of similarity, where it's basically the opposite, right? With that information, we build the phylogenetic trees, as Mike showed before. And there are a number of assumptions here, as Mike mentioned, a number of them. Because sequence is theoretically split into 2 descendant sequences, then phylogenetic trees are typically assumed to be bifurcating. From the origin, they keep bifurcating to the future. Topology is what we call to a branching pattern of a tree. So the pattern that we follow here, for example, this is a given topology that describes how this virus has been evolved and changing. And then taxa, for us, in this particular problem, are the virus sequences that we are using. Now, the problem comes, each of these graphics here are different topologies. So if 4 viruses are being compared, then there are 15 and 3 possible rooted and unrooted tree topologies, respectively. So if I have only 4 viruses being compared, there are 15 potentially different topologies, and only one of those is a true one, right? When the virus transmitted and evolved and changed, out of these 15 possible topologies, there is only one that is a true one if there are only 4 viruses being compared. Now, when we have only 10 viruses being compared, then the potential number of rooted topologies becomes 34 million. And we need to find out which of those is a true one. That's the problem that we have. So what we do is follow a process that is called reconstructing or inferring trees and phylogenies. And by that process, we try to identify, following certain principles and assuming certain things, as Mike described, what's the true topology. And that true topology is what we show as the phylogenetic tree that we obtain of those relations. But it's important to keep in mind that what we are trying to do at the end of the day is to identify the best model. And it's a model. It's not the truth. We are trying to identify what's the best description of what we are seeing in those sequences. Okay? So to do that, we follow different principles and different tests. Density, parsimony, likelihood. And all of them have in common that they ignore the association with certain epidemiological information, as Mike mentioned before. When we build those trees, typically we ignore or we just don't look at the location or the period of time where the sequences were collected. We just look at the sequences, not making use of this other extra information that we have. But intuitively, you may think that the place where the sequence was collected, the system, the type of farm, the time, all of that information should help us to identify what is the true topology, right? That should matter. And then we should try to take advantage of that information to identify that true topology. So one of the things that we found playing with this Bayesian analysis was that looking at only the pairwise distance in pairs is not a good predictor of the phylogeny. So it's basically what's showing here in the x-axis is just the nucleotide distance as I showed in the matrix. It's just the proportion of different nucleotides that you have in the data set. Whereas here in the y-axis is showing the result of a phylogenetic analysis that is showing actually the phylogenetic distance between pairs of sequences following a procedure that we'll discuss in a minute. And it's basically showing that there are a number of sequences that seem to be similar in terms of the nucleotide difference, but actually evolved in different periods of time, and may have not been related phylogenetically. So we can use phylogenetic methods, again, making use of a number of assumptions to infer the most likely time of emergence of the strain, the past probability of a spread between systems and between type of farms, and the population size, viral size in this case, which is actually the difference in the viral genetic composition. So Mike described this data set. We use almost 7,000 OR5 sequences collected between 1998 and 2015, 5 independent systems that were coded, type of farms that were, again, categorized at sites with sows and sites without sows. We selected those 174 Perez viruses using the maximum likelihood algorithm. So even though we refer to this as using the RFLP nomenclature, we use a maximum likelihood procedure to identify those viruses for the reasons that Mike explained earlier today. We ended up identifying a number of almost 300 sequences that seem to be related from each other between 2003 and 2005. Most of them were 174 that were obtained between January 2014 and March 2015. This is the slide that Mike showed before. And as he described, what we're showing here is the genetic diversity. So it's using the information to identify how different or how was the divergence of the virus population at different periods of time. And it's showing that it's an expansion in the diversity that then we use as a proxy for total virus population size. So this serves as an indicator, a proxy for the total number of viruses that are out there circulating. There was an expansion in the virus population size right before we started seeing those new cases of this particular strain. Then we use that information to link to the 5 participant systems that we have here. And we can see that initially the virus was sitting in one given system, here coded with C. And then apparently, just looking at this data, only 5 systems, it was introduced into this other system from where it spread and eventually affected their systems. Now, this information is susceptible of the systems that are participating and the quality of information that we have. That's why it's important, as was mentioned before, the swine health monitoring program and having enrollment and participation and sharing of data, because we improve our ability to make these inferences as we have more information to compare and to use. If we were to introduce a new system here, it may well be that we end up having a completely different conclusion. And then this is doing the same, but just looking at type of farms. So now we are looking at what we are seeing here is that the virus was mostly in sow farms, and then from the sow farms spread into other type of farms, which makes sense. I mean, we don't need this model to know that. But But when we found this type of with these things, then we gain confidence on the other findings that we are getting. If we would get the other way around, then you wouldn't believe me that much, really. So this is a video showing the results. Can we run this video, please? So what is showing, each of the dots there is showing the systems that were located in Alaska. And then what's showing the connections are showing the likely spread. Can we run it again? Showing the likely spread of the virus from each of those systems into the others, just based on the data that we had. The shades there are just, it's the inverse of the variance. So it's how certain we are that the virus was sitting there. So we don't really need to pay attention to that. But basically what we can do with this information, if we had information on the location of the farms, is to trace how a given virus is spread into different farms or different systems based on the genetic information that we have. So future plans. Again, this is extremely susceptible of the quality of information that we have and the amount of information. So we would like to demonstrate this to participants in the program and hopefully get more people eager to be part of this analysis. Hopefully that will help us to standardize and optimize protocols for data collection and sharing. We are looking for some plans to implement these ideas into analytical tools that could help to run this in near real time. The recently created Swine Health Information Center has some plans on that, and that may help to this day-to-day use of these ideas. We would like to provide near real-time interpretation of results. Certainly we will not be able to know what will be the new emerging strain, but hopefully with this information we can try at least to predict what's going to be the probability that the disease spread into other regions or other areas, and try to anticipate some actions, and eventually try to contribute to PRRS control at the national scale.